So we've discussed the chemical architecture within a neuron. So now let's go back to the mechanisms by which long-term changes in synaptic weights can occur and look at how these mechanisms are implemented by chemistry. Now earlier on we looked at this diagram. Remember that in this context, long-term means long-term relative to the duration of one postsynaptic potential injection, which lasts for a few milliseconds. Long-term changes can last for minutes, or hours, or indefinitely, and different mechanisms support changes on different timescales. As we discussed, fairly short-term changes result from increases in the effectiveness with which ion channels pass sodium ions, but the channels revert to normal in a matter of minutes. Somewhat longer-term changes result from increases in the number of sodium ion channels, but the channel concentration tends to return to normal within about an hour. Very long-term changes result from permanent increases in the size of the synapse, allowing more ion channels at normal concentrations, and these changes can last indefinitely. Finally, it's also possible for synapses with no ion channels to acquire channels. So, how does this work at the chemical level? Well, one key factor is that there's two different types of receptors for glutamate. The AMPA receptor is a simple sodium channel. The NMDA receptor is a calcium channel. It requires glutamate to open, but it also needs depolarization of the local membrane potential, while the glutamate is still bonded. If both glutamate bonding and depolarization occur at the same time, the channel opens to allow passage of calcium ions, and among other things, these calcium ions can act as second messengers, leading to the synthesis of more AMPA channels. And the larger number of AMPA channels increases the sodium current that flows when the next action potential arrives at the synapse. In other words, it increases the effective weight of the synapse. Most synapses have some receptors of both types. But silent synapses only have NMDA receptors, so they have no weight. But they can acquire weight in the right circumstances. So let's have a look at the process using the conceptual drawing of a synapse that we were using earlier. There's AMPA sodium channel receptors as before, but there's also NMDA receptors. And in an actual synapse, NMDA receptors tend to be located centrally in the postsynaptic density, with AMPA receptors in a ring around them. So if glutamate is released, it binds to both types of receptor. The AMPA receptors open immediately, and that allows a sodium current to flow into the postsynaptic neuron, and this is the postsynaptic potential injection. But the slight change in membrane potential because of this postsynaptic potential isn't sufficient to open the NMDA receptors. Chemical processes are constantly clearing glutamate away from the cleft, and the binding to AMPA receptors only lasts for a short while, so the AMPA receptors rapidly close again. But the glutamate stays bonded to the NMDA receptors a bit longer. So for a short while after the postsynaptic potential injection has been cleared away, the NMDA receptors are still bonded to glutamate. If the postsynaptic neuron itself fires during this short period, an action potential is launched down its axon, but in addition, an action potential backpropagates into the dendritic tree. If this backpropagating action potential arrives while the NMDA receptors are still bonded by glutamate, they open. and that allows entry of calcium ions. These calcium ions diffuse into the neuron and trigger chemical processes that result in synthesis of more AMPA molecules. The new receptors are inserted into the synapse, so in future there are more sodium ion channels that can be opened by release of glutamate, so the postsynaptic potential injected by one incoming action potential is increased, so the weight of the synapse is greater. So that's a quick view of how synaptic weight changes lasting about an hour can take place. But as I mentioned, changes can last a matter of minutes or can be fairly permanent, and different chemical mechanisms support the changes on different time frames. One important factor is that even if there are more AMPA receptors created, they'll eventually diffuse away unless the synapse gets larger to accommodate them. Now, let's step back and look at things on a somewhat larger scale and see how the synapse can be made bigger. As we discussed, glutamate synapses are located on spines. Close to the spine, there's often a synapse from a dopaminergic neuron. But spines are usually way out on the dendrites, a considerable distance from the nucleus in the soma, where genetic information is stored. But at the base of the spine, there are ribosomes, where proteins can be synthesized if the right messenger RNA is available and activated. Now, what determines the size of the synapse? Remember that a neuron has a kind of skeleton made up of those microfilaments. If this skeleton grows, the neuron grows. There's a microfilament skeleton inside the synapse, and if it grows, the synapse will expand. And if it expands, more AMPA receptors can be accommodated permanently, so the synaptic weight is increased long term. Microfibers are made up of long chains of a protein called actin, and actin can be synthesized from the appropriate messenger RNA. But extra messenger RNA for actin isn't usually available at the base of the spine. But if more of this messenger RNA is created in the nucleus and transported to the base of the spine, more actin can be synthesized in local ribosomes and transported up the spine and added on to the skeleton. That will expand the size of the synapse, and its synaptic weight will increase. Now, in reality, the picture is a bit more complex than this, because there's actually a dynamic equilibrium maintained between delivery of new actin into the synapse and breakdown of existing skeleton, which is then replaced by new actin. And generally, the result is homeostasis, with the synapse remaining the same size. But there can actually be short-term expansions in synapses, pushed by a higher rate of actin synthesis and a lower rate of degradation, that return to normal size fairly rapidly, unless the rate of actin synthesis is permanently set at a higher level by the arrival of new messenger RNA. So the simpler picture I described is adequate to understand what's going on.